Swayam Prabha. Digital India. Educated India.
Hello everyone, welcome to the course on computer aided drug design. In this class we are going to look at some simple drugs, and we are also going to look at some databases which gives a large number of structures. They are very useful for preliminary screening of molecules. As I showed you in the first class, these are some of the databases which contain structures of a large number of molecules. The zinc database contains 35 million compound structures with some properties we can buy if you want. Then we have this drug bank and drugs.com. These databases contain structures of drugs that are currently available in the market, over the counter drugs, FDA approved and so on, actually biotech drugs and so on. So if I want to know what is the drug available in the market for a particular disease, the first step is I will go into this drug bank and drugs.com. In addition, we also have the ChemSpider, ChemDB, PubChem, ChemMBL. All these contain structures of large number of molecules, searchable, and contains properties of all of them, different types of properties, molecular weight, solubility and so on, actually. So we will look at some of them in this class. Okay, the most well known, widely used drug is aspirin. Okay, it is called acetyl salicylic acid. So we have this the acid, so we have this salicylic acid. This is the acetyl group that is present here. Okay, this is used for pain, fever, so it is very widely used, and nowadays it is even being prescribed for reducing the viscosity of the drug, viscosity of the blood. So it is called a blood thinner. So aspirin is widely used. Some of the targets for aspirin is prostaglandins, okay, prostaglandin synthase 1, okay, then prostaglandin synthase 2. So this is called a cyclooxygenase 1 and cyclooxygenase 2. These enzymes are reductase type of enzymes. Okay, this gives you the ligand efficiency. And these are some properties of this particular molecule. Okay, they are called properties, they are called descriptors, structural features. So we call them different names: features, properties. We also call descriptors. We will use this quite often. Descriptors, just like if you take a human being, the properties could be height of the person, weight of the person, color of the skin, color of the hair, color of the eyes, so many different properties we can have. Similarly, for each chemical structure we can have large number of properties. We are going to talk about them later, okay, but molecular weight is one easy property which you all know about it. It has got two rotatable bonds. So we will talk about these properties later. Now let us look at another one, metformin. Metformin, again everybody nowadays know about metformin because diabetes has become a serious problem globally as well as quite a lot in India, and it is widely used drug for controlling diabetes. Okay, so this is metformin structure. It has got a lot of nitrogens. Okay, so it is highly polar, so it is highly hydrophilic. Hydrophilic means it is highly water soluble, as against hydrophobic, which is lipid soluble. So hydrophilic means it is more water soluble, hydrophobic means it is more lipid soluble. Because of the presence of nitrogens, it is highly water soluble. Similarly, if you have a lot of OH group, hydroxyl group, it will become highly water soluble. So this is metformin used for diabetes. Again, a lot of properties of metformin are given here. We will spend more time later as we go along. Look at this. These are antibiotics. They are called beta lactam antibiotics. These are broad spectrum antibiotics. Okay, it is highly available in the market. Penicillin, one of the oldest antibiotics, it was given during World War II for soldiers who had infection. Many soldiers before the advent of penicillin used to die because of infection, and the penicillin was the biggest boom. Okay. They are called beta lactam antibiotics. There are many beta lactam antibiotics. So, this is called a beta lactam ring. Okay. This is called a beta lactam ring. The 4 members cyclic with a nitrogen and a ketonic group, and many antibiotics which come under this class of beta lactam antibiotics will have this particular ring. Okay. So, we can have different substitutions. Here I have put R, we can have different substitutions here, but this particular ring is kept intact, that is called the beta lactam ring. 4 members cyclic with a nitrogen and a ketone. So, this is cephalosporins. Again you see this ring, the beta lactam ring, and then here the different types of R groups are put in. So, we get large number of cephalosporins. Okay. So, they are antibiotics, broad spectrum antibiotics, and there are many types of beta lactam antibiotics available currently in the market. Okay. So, that is the beta lactam ring. Okay. So, look at this antibiotics, amoxicillin. So, it has got this beta lactam ring. Okay. Penicillin 5, it is a oral, this one. Penicillin G. So, this beta lactam ring is intact. So, there are a lot of modifications to the side chains to improve its solubility or maybe improve its stability, to balance the hydrophilic and hydrophobic nature of the drug. So, many reasons they add these extra substitutions to improve the drug likeness property or maybe to improve. Okay. So, this is the general penicillin. As you can see here, the beta lactam ring is there, but there are so many different types of penicillin, penicillin G, penicillin V, amoxicillin, but all of them have this. Okay. So, how do they act? They prevent cross linking between proteins and hence cell wall synthesis. So, the bacteria cell does not grow because they prevent the cross linking of various proteins that are involved in the cell wall development. So, look at this combination of amoxicillin and clavulanic acid. Okay. They are used for treatment of bacterial infection. Okay. What happens is these bacteria became very smart and they started producing something called a beta lactamase enzyme. Okay. That is called a beta lactamase enzyme. So, that enzyme produced by the bacteria started breaking this beta lactam, thereby the antibiotic becomes ineffective or it loses its activity. The bacteria started developing this particular enzyme, and this enzyme will break this structure. So, the antibiotic loses its activity. So, what happened was they started introducing combination. One is called clavulanic acid, amoxicillin. So, this clavulanic acid will go and bind to this enzyme beta lactamase, thereby that enzyme is not able to do any job, whereas this antibiotic will start working and thereby it will kill the bacteria. So, this is called a combination therapy. Okay, this is called a combination therapy where we give 2 drugs, one drug acts on one target and another drug acts on the other target. Okay. So, that is called a combination therapy. Because the beta lactamase enzyme started degrading the beta lactam ring, they started giving 2 drugs in combination. So, this particular clavulanic acid will go and bind to the beta lactam enzyme, that is a beta lactamase inhibitor, whereas the amoxicillin will start working on the cell wall, thereby the bacteria will get killed. Okay. So, nowadays these bacteria become resistant, they are called resistant bacteria because they start doing different tricks. One approach is to produce these type of enzymes which will go and kill the or destroy or degrade the drug. That is one approach. Other approach is to produce something called efflux pumps. That means whatever drug comes inside the microorganism or cell is thrown out, that is effluxed out. Okay. So, that is called efflux pumps. So the bacteria started developing these type of new tricks, thereby they become resistant, and nowadays you must have been hearing about resistance bacteria, resistance tuberculosis bacteria and so on. So these are the tricks they follow, and so drugs are given in combination to overcome some of these problems. Aspirin, okay, we all know aspirin, there are different types of aspirins as you can see, phenacetin, then Tylenol. So all of them have different type of structural features as you can see here. Ibuprofen, it is also called Advil. These modifications are done in the structures, as I said, to improve the drug likeness property or to improve ADME or to make the molecule more stable or reduce toxicity and so on. Actually, we will look at some of them as we go along. Antihistamines, again all of us must have taken antihistamines. These are taken when to overcome swelling or redness, inflammation, sneezing, runny nose, watery eyes. So how do they act? They block histamine release from histamine 1 receptor. Okay, so they block histamine. When it gets released, you end up having swelling, itching, inflammation, runny nose, watery eyes and so on actually. So they are called antihistamines. Okay, they block the histamine release. This is what is called a histamine. This is the structure of the molecule. That is called histamine. So these are some of the antihistamine drugs which are in market: diphenylhydramine, chlorpheniramine. So we will see that they have the nitrogen groups here. Okay, that is why this ending is amine. Okay, so they have all the nitrogen groups here, that is why it ends with amine. These are all antihistamines used for allergy type of situations. More of antibacterial. These sulfa drugs were discovered very early. They have this sulfur groups, that is why they are called sulfa drugs. So as you can see here, sulfanilamide. So they have the NH2, so there is an amide group here. Okay, that is why it is called sulfanilamide. Okay, this is an aniline type of group. So different types of sulfagonadine, sulfathiazole. This is called a thiazole structure. A 5-membered ring with nitrogen and sulfur is called a thiazole structure because thio means sulfur, ZOLE is nitrogen. When you have thiazole, it means it is a 5-membered, nitrogen and sulfur is present. So you see different types of modifications to sulfa. These drugs nowadays are not used much because the bacteria have become more resistant to these drugs. Okay, and the whole world has shifted towards different types of antibiotics, like I showed you penicillin. Again, penicillin also has become very meek, bacteria have become resistant to that. So the medical fraternity has moved into much more powerful antibiotics. So there is always a war between the microorganism and scientists. As the scientists discover new drugs, the microorganisms somehow get resistance, and either through gene modifications or through design of efflux pumps or design of new enzymes to degrade the drug. So again scientists start discovering new molecules and so on actually. So these are sulfa drugs which are generally not used nowadays. Maybe for some of the skin infections it is still being used in ointments. Then other antibacterial drugs, you can see tetracycline, you must have heard all these names, aromycin, okay, again terramycin, fluoroquinolin. So these drugs, they bind to the bacterial ribosomes, whereas fluoroquinolins bind to the DNA replication. So the DNA does not get replicated. Okay, so you can see different type of mode of action of antibiotics, antibacterial drug. If you look at penicillin, they prevent cell wall synthesis. If you look at these type of drugs, tetracyclines, they bind to bacterial ribosomes. If you look at fluoroquinolins, they prevent the DNA replication. Okay, so pharma companies look at different possible targets and start designing molecules towards those targets. Ibuprofen, okay, this everybody would have taken because of inflammation and so on, in the joints, in the knee and so on. So, these are called non-steroidal anti-inflammatory drug, okay, NSAID. Once upon a time steroids were given if there was swelling in knees or joints, but steroids, as you know, has a lot of side effects. So, drugs were discovered, small molecule drugs were discovered to overcome this inflammation as anti-inflammatory molecule. So, they are called non-steroidal anti-inflammatory drug, NSAID. So, how does it do? It reduces the hormones that cause inflammation and pain in the body. They go and bind to some enzymes which produce prostaglandins, okay, that is how they act. Okay. So, large number of drugs which we use very commonly I have shown, which is very important for us to know the structural features. We need to know the structural descriptors which are normally describe the molecular properties. So, that is called cheminformatics, which tries to find out the structural features or structural descriptors of molecular structure. So, let us look at some databases like I said before and see how we can make use of them. Okay. Okay, let us first look at this database called Zinc 15 database. This database contains structures of a large number of molecules. As you can see, 100 million, it keeps going up every day. Purchasable, that means if you think you have found a good possible lead compounds based on in silico, you can even buy it, okay, that is the beauty of it. Now, this Zinc database, how do I go about searching? I can do click on this. So, search for a substance. So, I can type say for example aspirin, yeah. So, we find structure, I click on it. So, it gives a lot of information. This is the structure. As I said, aspirin is nothing but acetyl. This is the acetyl group. This is the salicylic acid. So, it has got a benzene and an acid here. It is available in stock and so on. Okay. The net charge on the molecule is -1. Why does it get to -1? Because the OH can easily dissociate. So, you can get a O -1. It does not have any hydrogen bond donors, okay, because it gets the O and H gets okay, dissociated as O minus. So, hydrogen bond acceptors, that means oxygen and nitrogen are called hydrogen bond acceptors. So, 1, 2, 3, 4. So, it has got 4 hydrogen bond acceptors. TPSA means total polar surface area. The PSA means polar surface area. Okay. Polar surface area, it has got 2 rotatable bonds. So, 2 rotatable bonds it has got, then okay, and so on. So, a lot of properties it is giving, and then in addition it tells which enzymes it goes and binds to. If you click on this, we can find out more details about those enzymes. Okay, as you can see, these are other analogues of this, okay, different okay, analogues of this, as you can see here, different analogues of aspirin. And then what has been done, has any clinical trials been done? The details are given, phase 4 clinical trials, antiplatelet therapy. So, as I said, aspirin is nowadays being used for thinning of blood. It also used for preventing platelet aggregation. Okay, so strokes, it is also being given now for strokes also. So, phase 4 clinical trials have been done. So, it is in the market for treatment of blood thinning as well as for stroke. It is warfarin or aspirin is being looked at it, this term completed. Like that, you know, what are the clinical trials that have been done with the aspirin, what is the current status, 2005, 2006, 11, 7, 11. So, those details are also given on this, and so good amount of information is given about this particular molecule. Okay. So, if we are interested to buy aspirin, we can always get it here, we can write to them and also get these information. Okay, let us look at another structure, metformin for example. Metformin, you have metformin here. So, if you click on that, okay, so this is metformin. Again lot of properties of metformin. Net charge is 2, because see, you can see plus charges are there. So, it's 2 hydrogen bond donors, it has got 1, 2, 3, 4. So, 4 nitrogen groups which are hydrogen bond donors, there is no acceptors. Okay. And lot of information about it is being given. Metformin and rostroglizone, this is also another anti-diabetes drug, okay, for treatment. So, some clinical trials details are being given here, okay, and then it's some of the properties of this molecule, and then what type of studies are being done in clinical trials. So, all these are given. So, if I can click get more information on this, okay, yeah, so I can get more in. So, this is a very useful database. Okay, in addition we can do a lot of things here. We can draw structures here. For example, imagine I am drawing a structure here, okay, I can draw a chemical structure, and then okay. Okay, so I am interested to look at molecules in the database which has got this substructure. So, I can say substructure search. So, I can draw different substructures here and search this database to find out whether there are any main structure with this type of substructure present. So, because this is a very common thing, it is searching and searching, it will take a long time, but if I have a big substructure and then ask it to search, I can do that. So, I can draw different structures using this particular template. You know, I can draw benzene rings, 5-membered rings, heterocycles, carbon, nitrogen, oxygen, sulfur, different as you can see, chlorine, bromine, like that I can draw. If I know the name, of course I can give the name. If I know, oh, look at this. So, it is found so many molecules in the database which has this particular substructure. As you can see, this benzene ring with the O, that is what I wanted, right. So, it is found huge number of substructures, huge number of molecules with this particular substructure, okay. I can take it further and do some studies, or I can look at a much more stringent filter and try to reduce them. As you can see here, a large number of molecules have this particular substructure. The benzene ring, there is an oxygen, there is a in the para position there is substitution groups. As you can see, more and more molecules are coming up. Imagine I just click on one of these, yeah, so more molecules are there. Imagine I just click on this particular molecule, okay. So, it gives you the details of this molecule, okay. This is the molecule here, okay, and this is the zinc ID of that molecule, and then some properties you can find here, log P. We will talk about all these. Molecular weight is given here. We can buy this molecule from them if we think it is very okay. Not much information about this molecule is there. So, okay, that is called a substructure search, okay. So, the beauty is we can do that sort of search, okay. In addition, there is another search which or if you know the zinc ID, that means this is called a zinc database. So, if you know the ID, we can do that also. Okay. So, you can draw the structure, that is the beauty of it. Now, let us look at drug DB. Okay. So, there is this drug bank which contains large number of drugs that are currently in the market. So, it contains 10,508 drug entries, that means these are approved by FDA and they are in the market, including 1738 small molecule drugs, so many biotech drugs. Okay. So, all these are there. So, now we can start searching this also, for example, okay, this is a drug of month, drug bank top drugs, acetaminophen, this is used for aches, pains, acetylsalicylic acid, metformin I talked about, aspirin I talked about, morphine is a drug used for severe pain, it prevents, it reduces the pain which you have. Okay. So, this is a very useful database we can use for our study, and you want to identify some new drugs. Okay. So, we can say again metformin. Okay. So, we can search for metformin. Metformin is a biguanide. So, okay, it gives you all the details about this particular drug. Okay. So, look at drugs currently looking for. Okay. Okay. So, I typed something called beta blocker. This set of drugs are given for cardiovascular problems, especially at high blood pressure. There are certain beta adrenergic receptors which are blocked by these type of drugs. So, large number of drugs, they all end up with this particular title, bupranolol, indenolol. So, all these drugs come under this category of beta blocker. This is non-selective beta blocker. So, normally this is given for high blood pressure. For example, let me click on one of them. Okay. So, it gives the structure of this molecule, and then approved, it is a beta blocker. Okay. And then international other brand names, because companies sell it as brand names. Okay. What is the molecular weight, chemical formula, then pharmacological details. Okay. What is the pharmacodynamics, and how it acts, how does it bind to, and so on actually. So, a lot of information, of course details about drug interaction, how it is interacting with other drugs, because nowadays doctors may prescribe 2-3 drugs. So, how do they interact with other drugs, and so on. Then clinical trial details, then some experimental properties, melting point, water solubility, and then these are predicted properties. These are experimental, these are predicted properties, solubility, okay, pKa, that is dissociation, acidic, and so on actually. Okay, lot of information is being given, number of rings. Bioavailability is a very important parameter. It tells you when I take a drug orally, how much is actually available at the target site. Okay. And so on actually. So, it gives lot of information. Then as you can see, predicted, that means computationally predicted ADME properties, adsorption, distribution, metabolism, excretion, T is toxicity. So, it gives you information about using software. I think they predict lot of properties. As you can see, different types of properties are being predicted, and they are all given in this. So, if I want to do some analysis studies, these type of information is very useful. So, drug DB is another useful database. Then we also have as I said another database, that is the drugs.com. Okay. So, let us look at that also. Okay. It gives you a lot of information about different types of drugs that are currently in FDA approved. Okay. So, we can do a drug index based on condition, class, generic drugs, over the counter drugs, international drugs, natural products, veterinary products, drug side effects, Okay. So, if I am going to start a research on a particular disease, first thing I will do is look at DB and drugs.com. So, I will know all about currently existing molecules. So, I can know their properties, I can know their mode of action, I will know how they act, what are the side effects. So, that is a very good step to start from. Okay. So, you need to start from that particular step. Okay. So, other databases, let us look at drugs.com, and so you also have this ChemDB, that is another database. Okay. So, ChemDB is a chemical databases online. Okay. So, you can see this is a chemistry databases reference. We can search by structure. So, I give the structure, and then so we can look at different types of structures, and then we can start looking at. Okay. So, this is a bioscreening. So, it gives we can start looking at structure search. Then we also have the PubChem database, which gives you information on various types of this is called a PubChem database. Again, this has got a large number of structure searchable options. We can give property searchable options, 3-dimensional structures searchable options, and so on. So, we can say compound aspirin, yeah. So, different types of aspirin you can search. Okay. So, look at this, it gives you a lot of information, it gives you the IUPAC name, that sort of things. Or we can, if I want to know some bioassays, how do I okay. So, it tells you some assay for aspirin, various types of assays for aspirin, either to detect aspirin or look at assays for its interaction with different targets. So, that sort of biochemistry related information we can do. Again, this also has a structure search option, so we can draw the structure. Okay. Search substructure, structure. Okay. So, we can draw structures here, as you can see here. Okay. A lot of things can be done. Yeah. So, I can do a lot of things with this. Okay. So, we can look at this, and then we can say okay, draw a structure, edit, search, yeah, it is waiting, query, yeah. So, it is doing something. So, it will look at substructure search for molecules with that particular substructure. So, this is called a PubChem database. Okay. So, you people can start playing with all these types of different databases available in the web. Okay. All these are very useful databases because it gives you structures and a lot of properties which you can make use of for doing different types of calculations. So, because we will not have enough time to go through each one of the database in detail, I would suggest that you people go through all these databases. We can look at it based on the name of the molecule or the drug. We can draw structures, and also we can do a substructure search, and we can do based on properties. We can do search based on disease conditions. Okay. So, a lot of studies, data collection and data analysis can be done with these type of databases, and we will be using some of these databases as we go along. Okay. Thank you very much for your time.